Well, I will use the next 5 minutes to show you that in addition of drugs, small molecules, and biologicals, you could use cells to treat patients. So the idea is cell therapy basically means using cells. And we have been using cells for the last 25 years. The bone marrow transplantation is a cell therapy, and it works. It works quite well. So in addition of bone marrow cells, perhaps we could use other cell types. And I will focus in diabetes. Thanks for the previous presentation, so I could go faster. But you can use the same examples for other autoimmune diseases, for neurodegenerative diseases, for a long list of rare diseases, etc. So let's go into the first question. Can we convince stem cells to become insulin-producing cells and use these cells to substitute for the cells that diabetic patients don't have? So our group was the first to convince mouse embryonic stem cells to be insulin-producing cells. When these cells are injected into a diabetic mouse, it drops the blood glucose. So it seems that we had a solution. When we move into the human ES cells, we realize that the problem is much more complex. Mouse are not humans. And in the case of convincing a human ES cell means a long list of factors that those who have a star are factors contributed by my group and in our publications. But basically we're going to a cell and still looking for new factors that could activate sirtuin-1, etc. And let's go to these results. So if we can see, well, probably it's not clearly seen, excuse me. In the second, well, we have a way to increase the insulin content, and the best way to show that is that we have both insulin and C-peptide, the connecting peptide between the two branches of insulin. When the cells, human cells, are injected into a model of immunocompetent mouse that can receive human cells, normalize also blood glucose. But how far are we from a treatment? We are far away from a treatment. Why? We need better cells. But in addition of having better cells that do behave like the adult human cells, we have to solve the origin of diabetes, the autoimmune attack. So stop the autoimmune attack, solve the cause, put the cells. It will take years, although there is a group, a company in California that is now in negotiation with the Food and Drug Administration to use a similar type of cells and put the cells in a hollow fiber in such a way that the hollow fiber is put in the subcutaneous position, so you can remove the hollow fiber in the case that the things go wrong. But meanwhile, can we do something for diabetic people? So perhaps not to cure diabetes, but to ameliorate the condition of diabetes. And I will focus in a condition which is the diabetic foot. Some patients develop vascular and neural problems that make the feet to become gangrenous, so they lose the feet. And the solution is amputation. And the mortality is very high around this condition. And we recently published that using autologous cells that from the same patients that we could separate and inject into this leg could stop amputation and increase the vascularization. And now we are running, I am running 3 clinical trials using different cell types and trying to see which cell type or which combination is better in order to improve the situation. So these are cells doing beneficial effect for the patients. And this is a long list of clinical trials done in Spain now, thanks to an advanced therapy program that I personally put while I was in the cabinet. But most of them, except for one, are investigator-driven research. And investigators don't think about business, about, okay, I do the proof of concept, but we have to move into phase 2, into phase 3, but that costs a lot of money. Who has the money? No one has money. And that's the next point, which is the business model for cell therapy. And in fact, we cannot take the example of the big pharma model. So the presentation of Dr. Sands was very clear in the case that probably we could discuss whether it's the innovation model, what we are discussing, or the pharma model, in fact. So we can't change it. And it's the healthcare model, because pharma is only a part of the healthcare system. And perhaps we have to reshape not only the health system or the healthcare system, but also how pharma plays a role in the healthcare system. Obviously, we cannot spend 1 million euros to develop a new cell therapy, or so we have to reduce costs, to reduce time, and think in the market access and in the reimbursement. But the regulations for cell therapy are as strong for the small molecules. So I recently reviewed the regulation in Europe, and I was surprised how many regulations we have in Europe, and still increasing, and how many examinations you have to pass. So where is the model? And until a few years ago, we thought that we had two models. Autologous means the same cells of the patients, you make a treatment and give the patient back. So that's a service, that's not a product. Okay. The allogenic model is we take cells from a donor and escalate the number of cells, and we can have off-the-shelf drugs. Okay, perfectly, so we have the models. But Dendreon put an autologous treatment and put a product on the market that was approved by the Food and Drug Administration, and the value of the company increased suddenly. So probably we don't have a clear model. Is the field mature enough? This field is not for the last 10 years. I mean, it has been for the last 50 years, probably. We are in the second half of the Gartner cycle, so we're mature enough to go into the, not only market, there are plenty of examples of marketing regenerative products, but in addition to that, probably some products are not commercial, and just to end some considerations. So in the past century, we were used in small molecules. While I was a student of medicine, pharmacology means molecules. So I was trained to use molecules. Later on, biologicals appear. So monoclonal antibodies, but that's slightly different from molecules. And now we are talking about cell. But a cell is not a molecule. A cell is a living organism and has more than 4,000 molecules within. So what can we do to understand? Not only, in the case of small molecules, we could know the target, we could talk about pharmacodynamics, we could establish a dose 3 times per day, okay, and we could follow the toxicology, etc. In the case of biologicals, differs. We use a monoclonal antibody once a week, once a month, once every 3 months. What happens with the cells? In the case of the cells, the cell interacts with another cell. And I have recent results and published results in which we could show that the phenotype of the cell changes when it enters into the patient's blood. So what happens with a molecule is not metabolization, it's changing the phenotype, it's changing the behavior, it's interacting with other cells. It's trophic, it's anti-inflammatory. So we have a model that we have to define in order to be able to innovate and also to introduce these drugs, because from 2003, cells used in therapy are drugs as well. But we don't know. When the regulatory agencies ask me about biodistribution, about toxicology, about pharmacodynamics, which is the answer? The second, will I be able to reach phase 3 and registration and put a new treatment if we don't have ways? Is the patent system good enough for cells? How can we cover the intellectual property? And the situation now, and that's the end, is that I am a founding member of the Regenerative Medicine Coalition. That's 3 centers in Europe, 3 in the States, and 1 in Canada. I am also running a program in Europe to check for the bottlenecks of this process. And the first conclusion is that everyone is doing everything. And that probably is not the solution. Thank you very much.